Hello students, welcome to EPG Pathshala. I am Dr. M.N. Gupta, Emeritus Professor from IIT Delhi. Today we are going to talk on the module Vaccines from paper Immunology. This is the first module on vaccines. Control of infectious diseases requires multi-pronged efforts. Animals which are carriers have to be quarantined. Improving quality of water supply, sewage discharge methods, and educating people about value of personal hygiene are all important measures. Vaccination, the induction of immunity, is part of this. So the objective of this module is to understand requirement of a good vaccine and to understand in this context active immunization and passive immunization, to learn about various types of vaccine designs, and to learn about merits and demerits of the use of live and killed pathogens as vaccines. So the concept map looks like, essentially it's all about vaccination, two kinds of a vaccination, one involving passive immunization, another involving active immunization. As far as active immunization is concerned, it can be carried out either via by use of the live pathogens or by using the killed pathogens, and the various types of vaccines and how do we administer them. Let's not forget that immunology started with vaccination. But design of vaccines does not have just historical importance. It's a long pursued aspect of immunology which remains a frontier area even today. We now have few successful vaccines. Eradication of smallpox from all over the world and polio from most parts of the world is a major trump of vaccination. Yet, there are many diseases for which we still do not have vaccines. Let us look at what has worked and why it has not worked with some diseases. Jenner used vaccinia for providing immunity against smallpox in humans. The word vaccination was used by him, and Louis Pasteur continued its use while developing such materials for creating protective immunity against few other infectious agents. That is how immunology started. It is interesting to note that in 1982, WHO announced global eradication of smallpox. So what started with Jenner culminated in 1980. Vaccinia worked as it only persists briefly as a subcutaneous infection. It nevertheless stimulates immune responses cross-reactive with smallpox virus antigens. The broad approaches followed subsequently for developing vaccines were: number one, to search attenuated pathogens with reduced pathogenicity but with capacity to stimulate immune responses. To use killed organisms, these are safer as there is no risk of systemic infection, especially in immunodeficient and immunosuppressed individuals. Also, to try to use purified components which are effective immunogens. Effective vaccination is available in the case of large number of diseases which include measles, mumps, polio, tetanus, diphtheria, whooping cough. In many countries, children are advised and in fact required to be immunized against these diseases. It is a growing list actually. This slide begins to address the issue of what is a requirement for designing a successful vaccine. The first concern, of course, is the choice of the microbial antigens, if they are going to be the basis, which is quite often the case for designing a successful vaccine. Which microbial antigen then is the best immunogen? What is the point of entry of the infectious agents? Generally, that is the site where vaccine is best injected. What are the immune responses which needs to be manipulated? For extracellular infectious agents, antibody provides a good defense. Where intracellular infection is involved, effective CD8 T cell response should be induced. Thus, each issue presents some challenges. Infectious organisms which have large number of serotypes, for example, Streptococcus pyogenes and Streptococcus pneumoniae. In these cases, a large number of antigens would be required to be used together. Often correlation between protective effect and a particular antigen or its mechanism of action is missing. In some early vaccines of pertussis, there have been occasional reports of brain damage. In such cases, mechanism of protection or some side effects even are unknown. Nevertheless, whenever the effective antigens have been identified, these are preferable to using killed microbes which have multiple antigens and may cause problems such as hypersensitivity. Also, antigens can be produced more economically these days through recombinant methods. There are some examples which show that the mechanism of protection by the vaccine is known. This is one example which illustrates how that is sometimes described as a basic research pays dividends by providing insight into a medically relevant phenomenon. Some local sites such as genitourinary tract or gut require immunization at the site of infection. For example, intravaginal vaccination with Neisseria gonorrhea is to be preferred over systemic route. Similarly, oral vaccine for Vibrio cholera is believed to be more effective than injected form. In this context, let us recall whatever we discussed about passive immunization. Passive immunization consists of injection of preformed antibody intravenously or intramuscularly. These antibodies are derived from animals who produce high titer of these antibodies to specific microbes. Passive immunization is useful when a rapid protection is required, such as in the case of diphtheria, rabies, some Clostridium species or hepatitis B. Passive immunization is obviously useful for immunocompromised individuals as their immune system cannot offer protection. IgG class antibodies are generally injected every 3 weeks to keep their effective level in the body. Horses are generally used for obtaining these antibodies. Unfortunately, repeated injection carry the risk of antigen antibody complex formation and serum sickness. The challenges in designing oral vaccines involve ensuring that those reach the target epithelium or lymphoid tissue and generate limited infection. It should be a stable mutant which results in the protection and does not revert back to the highly pathogenic form. It should have only limited replication cycles so that it has only limited proliferation, just enough and adequate for provoking immune responses in there. Yet another important factor is that the vaccination with oral routes has a higher frequency of resulting in tolerance. Let us begin to look at the various types of vaccine designs. First on the list are toxoids. Toxoids are obtained by modifying toxins chemically such that it retains its immunogenicity but abolishes its toxicity completely. The extensive literature on chemical modification of biologically active proteins and bioconjugation chemistry is of considerable help. Early immunochemists were actually excellent protein chemists, and that has led to many early successes. The second design is attenuated live vaccines. These are bacterial or viral preparations modified to eliminate their pathogenic property. In general, these provide better protection. However, as these may proliferate, immunocompromised people will have a problem. Subunit vaccines consist of an antigenic component isolated from the infectious organism. This is protein in nature and hence can often be obtained by the recombinant route. Vector vaccines are antigen genes inserted into a non-pathogenic viral factor such as vaccinia or adenovirus. Then we have a conjugate vaccines which are essentially weak antigens which are coupled to a toxoid. The toxoid stimulates T cells which help B cells producing antibody against the weak antigens. An example is 3-polysaccharide antigens consisting of meningitis C organism coupled to a diphtheria toxoid. DNA vaccines so far have not been approved for use in humans. Here the DNA responsible for producing suitable antigen is taken up by the cells. The last design consists of what are known as a combined vaccines. Many vaccines are given together in infants. The trivalent DPT vaccine for diphtheria, pertussis and tetanus is the most well-known example. UNICEF has also recommended a pentavalent vaccine which in addition to these has vaccines for hepatitis B and Haemophilus influenza B. In addition to adjuvants, vaccines may contain stabilizers and preservatives. In developing countries like India, absence of a cold chain, which means basically keeping the biological sample at cold temperature to prevent loss of its biological activity or property from the production point to the administration point. So The absence of a cold chain right up to remote non-urban areas is a big handicap. Stabilizer and preservators can solve this problem partially and are definitely not substitutes for the cold chain. An important issue obviously is the administration of vaccines. Most vaccines are administered in the form of subcutaneous to intradermal injection. Some oral vaccines are available. Polio vaccine of the Sabine type and rotavirus vaccines are examples of oral vaccines. Nasal route has been used for flu vaccines. Nanoparticle vaccines show promise for being delivered through mucosal and systemic route. We had earlier discussed adjuvants as materials which, when administered along with an antigen, enhance immune response. Some bacterial products which are used as part of vaccines designs have the following composition. BCG, which is Bacillus calmette-guerin, is a non-virulent strain of M. bovis. Muryl dipeptide, normally simply called MDP, is the cell wall component which has adjuvant action. Lipopolysaccharides, part of gram-negative bacterial cell wall, helps in B cell proliferation and causes macrophage activation. Bordetella pertussis toxoid binds T cells and facilitates these proliferation. These adjuvants are part of the many vaccine designs. In the context of vaccines, nanoparticles in nanoparticle vaccines actually function as adjuvants. So in nanoparticle vaccines, vaccine is adsorbed on nanoparticles. While currently accepted definition of nanoparticles include particle sizes up to 100 nm, in many cases particles of 10 to 1000 nm have been described for such applications. The materials used for preparing such nanoparticles include polylactic acid and polyglycolic acid, polylactide coglycolide, which is called PLGA, nanoparticles coated with Menon target mannose receptors to APC. Monoclonals have been used to coat nanoparticles as homing devices for a specific target on dendritic cells. Apart from nanoparticles, liposomes, muramyl dipeptide, immune stimulating complexes such as containing cholesterol and phospholipids and bacterial toxoids show high promise for being approved for human use. Right now, only aluminum and calcium salts are approved as adjuvants for vaccine preparations. Other adjuvants being studied include interleukin 1, interleukin 2, and interferon gamma, which are all cytokines. The list also includes immune complexes and Freund's adjuvant. Inflammation caused by the injection brings in macrophages and dendritic cells to local sites. Consequent cytokine production leads to recruitment of T cells. Ideally, an adjuvant should be able to stimulate both cytotoxic and helper T cells. As has been mentioned before, vaccines containing live organisms do not need adjuvants. A broader term is, instead of adjuvants, is biological response modifiers, which mostly enhance immune response, and this list then includes adjuvants. Toll-like receptors and cytokines, for example, can be termed more properly as biological response modifiers, that is BRMs. The debate about pros and cons of attenuated pathogens versus killed pathogens has often been a bitter one. Polio vaccine exemplifies it best. Let us first understand what are these two types of vaccines. Jonas Salk produced killed virus vaccine, whereas Sabin's polio vaccine consists of attenuated live virus. The debate continues. Both types of vaccines have positive and negative features. In the first module on the history, the very first module, which was on the history of immunology, we mentioned the ancient Chinese practice of using scabs of mild cases of smallpox for vaccination. So in this approach, there is an inherent hazard of disease breaking out if the organism starts multiplying inside the body at a rapid rate. In crosslinks. Other examples are of alkylating agents for nucleic acids such as ethylene oxide, ethylene amine, acetyl ethylene amine, and beta propoliectone. Vaccines based upon this approach are only able to produce humoral immunity. Salk polio vaccine is given intramuscularly and uses formaldehyde for inactivation. Such vaccine preparations can be easily mixed with other vaccine preparations. Poliovirus has three major strains. Type 1 virus is more dangerous than type 2 and 3 and seems to be far more frequent cause of paralysis. However, there is a cross-reactivity of antibodies. So, infection with type 2 prevents paralysis chances upon infection with type 1. Immunity by killed microbes has shorter period as compared to attenuated vaccines. One belief among immunologists has been that there is no immunity like convalescent immunity. This relates to the observation of Jenner's days that those which survived the dreaded smallpox never got the disease again. It is true of polio. The viral infection gives lifelong immunity. One has to, however, remember that convalescent immunity is not absolute or permanent. Sometimes a different strain may crop up without enough cross-reactivity. In some cases, revaccination is required. A good example is flu vaccine. So, lasting immunity is not ensured in every disease. The real advantage is in the case of immunodeficient people with virulence totally eliminated. The killed microbe vaccine can be given safely in such cases. The killed poliovirus vaccine does not prevent colonization of intestine by the wild poliovirus. In general, whenever cell-mediated immunity is important, in those cases, the vaccines based upon killed microbes concept may be inadequate. Thus, if the natural defense mechanism against some microbes require other mechanisms to operate, simple antibody production by such vaccines may be a handicap. However, as the examples listed here show, enough protection is available in the large number of diseases. Production of antibody will elicit some B cell, TH cell cooperation. Salk polio vaccine does not lead to detection of anti-polio IgA in nasal and duodenal secretions. This has the disadvantage of not being able to fight the virus locally. Serum IgG may play some protective role, however, at epithelial surfaces. Tetanus, diphtheria, gas gangrenes, and cholera represent diseases which are caused by toxins produced by the pathogens. Bacteria produces these toxins to damage host cells, which allow invasion. Toxoids prepared by reactions of formaldehyde with exotoxins of diphtheria and tetanus bacteria are examples of the success of this approach. Toxoids are given along with adjuvants. In many cases, adsorption on aluminum hydroxide works well. Metal hydroxides are known to adsorb many proteins very well and in fact forms one of the method of enzyme immobilization. In case of cholera vaccine, the cholera toxin subunit B is conjugated to killed Vibrio and is better as it facilitates gut mucosa to form antibodies. Given current state of the art in protein production, isolating a pure protein antigen is a viable proposition. One advantage is that this avoids antigen overload from other antigens which are present on the pathogens that may lead to hypersensitivity reaction. The antigen for this to work must have an epitope which stimulates B cell, but it is also crucial to have an epitope which is able to recruit CD4 T cells. The approach benefits from the fact that extensive work on epitopes responsible for pathogenesis has been carried out in many cases. Currently, epitope identification has been adapted to the high throughput mode and can even be outsourced to some specialized companies. In herpes virus, glycoprotein D, which is called GlyD, results in protective CTL response. Effective delivery methods resulting in complete immune response remains a challenge to this approach. Vaccines with purified capsular polysaccharides have been designed for infections of S. pneumonia and Neisseria meningitis. The former was produced by Merck with Pneumovax 2-3 and by Lederle Lab with New Immune 2-3 as trade names. The disadvantage is that polysaccharides are poor antigens for class switching and producing B cell memory. Overcome this, a vaccine for H. influenza has been designed by linking type B polysaccharide to tetanus toxoid as a carrier. It did not produce memory T cells specific to the influenza infection. The first successful example of purified antigen approach has been hepatitis B virus surface antigens, which was produced by cloning the viral antigen yeast. Again, however, it does not produce TC response. Given the advance in protein expression by cloning, this would continue to be a very promising area in vaccine designs. This list gives the recommended vaccine schedules in some cases. Many vaccines which are now commercially available are accepted by the medical community worldwide are given to humans either routinely or just before travel, and people whose working occupational environment makes them more prone to infection with a particular microbe. Herd immunity is a useful concept in vaccination programs. If significant percentage of this population is vaccinated, it creates islands of disruption in the spread of infection. This eliminates the number of hosts wherein the pathogens may multiply. Both factors protect the non-vaccinated individuals from that pathogen. Now it is a standard practice to immunize children against pathogens for which safe and useful vaccines are available. In USA, for example, pediatric population is given vaccines for following diseases. Examples include diphtheria, tetanus, oral poliovirus vaccine, combined vaccine for measles, mumps and rubella, Haemophilus B diphtheria toxoid conjugate. In India, polio oral vaccine administration program has been launched few years back and is already showing very remarkable results. BCG vaccination programs are different in various countries. In USA and in UK, it is based upon risk perception. Incidences of more than 40 per 100,000 individuals are taken as indicator of high risk population. In some countries, BCG vaccination is mandatory. Not all individuals respond to vaccination in a similar way. 100% population thus will never be protected by vaccination. However, because of herd immunity concept, vaccination is still effective in controlling large number of cases. Immunodeficient individuals, individuals on immunosuppressive drugs, under extreme conditions of temperature or stress situations, and malnourished individuals all show poor immune response. It should be remembered that vaccination efficacy depends upon the individual's immune response. Epidemiological studies on a disease form the basis for designing vaccination strategies. Continuous monitoring with accurate record keeping are critical in assessing the success of any mass vaccination program. Immigration authorities of different countries require proof of immunization against specific diseases. Malaria infects nearly 1 million people globally. About 10% of these are serious cases. Malaria is caused by protozoan parasite called Plasmodium falciparum, which infects erythrocytes. In rodents, the malaria parasite is Plasmodium uvuli. Antibody-mediated mechanisms involve blocking invasion by inhibiting its attachment to a new host cell. Plasmodium species infect erythrocytes as merozoites through binding to a receptor. This binding is blocked by specific antibody. Opsonization by antibody also encourages phagocytosis of infected cells by macrophages. Malarial parasite evade immune response by antigenic drift. This parasite also releases soluble antigens. These are polymorphic and have repetitive amino acid sequences. These antigens act as decoy by multiple mechanisms. As a consequence of these properties of malarial parasites, a successful vaccine for malaria poses enormous challenge. In this lecture, we have learned the following. We learned what are the properties of a good vaccine. Good vaccine means the one which offers protection as well as it has practically no side effects or potential side effects or the danger of the disease actually breaking out in the vaccinated individual or population. We also learned that the vaccination, immunization can be active and passive. We have learned very early in this paper about these two modes of active and passive immunization, but we revisited them in the context of vaccination. There we talked about how the immunization can take place by the modes. Here the emphasis was that how do we in these two modes protect the individuals by introducing by active immunization mode or the passive immunization mode. So thereby that discussion led to our talking about various types of vaccine designs, toxoids and killed pathogens as vaccines were discussed. We have just discussed various types of vaccines. Many of these designs need to be looked at more closely. For example, we need to understand how live pathogens can be used safely as vaccines. After all, it all started with Jenner using a live virus. Then there are some new concepts like DNA vaccines. They show considerable promise. In theory, they look like a brilliant idea. However, yet till today, there is no DNA vaccine which has been approved by regulatory authorities to be released on the humans. So obviously there are some challenges, some hesitations. When we discussed Journey's theory of network selection, we had hinted that this concept of networks is not totally abundant. We will see in a subsequent module the application of this brilliant concept in vaccine design.